Good day, people. Welcome to One-on-One Maths Private Tutors. Today, we are going to look at this question that came in O-Level Cambridge Combined Science, the alternative to practical paper, paper 6. Now, let's look at our papers first. Okay. So, in combined science, either you take the core part or the extended part. So, if you take the core part, you write papers 1 and 3. All right, you write paper 1 and paper 3. If you take the extended part, you write paper 2 and paper 4. But on top of that, because these are theory papers, on top of that, you either go for a practical paper 5, which is a practical test, or paper 6, an alternative to practical. So it is usually advisable for people who do not have lab experience to take the alternative to practical because it's theoretical. It's not practical. You write it theoretically, which is exactly what we are doing today. All right. So this paper has 40 marks. The other papers, paper 2 is, the first paper has 40 marks. The second paper is 80 marks. So we can see these marks are like a quarter of the whole exam. All right. If you take core, paper 1 is 40 marks, paper 3 it's 80 marks, and then you can take the alternative to practical for 40 marks. So bear that in mind. This is a quarter. We need to have practice on this. Right. But the papers basically are similar. If we go through 4-5 papers, you'll get an idea. Between now and November, if we can go through 4-5 papers, which is what we are starting now in July. All right, we start this in July so that by November we are ready for the alternative to practical. So you'll find in these questions, it's a combination. In one question, you'll find a bit of physics, a bit of chemistry, a bit of biology. It might be primarily a biological question, but you'll find a bit of the other subjects in it because it's alternative to practical for combined science. Okay, let's look at this question. This is primarily a biology question, but we will find a bit of physics applications and chemistry applications in it. All right, let's look at this. A student investigates an enzyme catalyzed reaction. So we can see when we're talking about enzymes, we are talking about, in biology, enzymes are proteins that act as biological catalysts. All right. Hydrogen peroxide is broken down by catalase. Okay, so we have chemical hydrogen peroxide, H2O2, to those who know it, is broken down by catalase. This is the enzyme found in living cells, such as the cells of potato tissue. Oxygen gas is released during the reaction. Okay, so hydrogen peroxide is broken down to H2O plus O2. So balancing equation you should know, but I'm sure it's not about this, but just to have the perspective to know what is happening. So the enzyme is catalase. In the presence of catalase, hydrogen peroxide is broken down to water and oxygen is released. Okay. Procedure: the student, step 1, labels 3 test tubes A, B, and C. So you have 3 test tubes. Label them A, B, and C. Okay. Make up 3 different concentrations of hydrogen peroxide solution by adding the volumes of 6% hydrogen peroxide and water as shown in table 1.1. Let's see what is happening in this table. Test tube A, we are putting 2 cubic centimeters of 6% hydrogen peroxide and diluting it with water of volume 4. So the percentage concentration now is 2. So it's coming from 6 to 2 because we are adding water into the hydrogen peroxide solution. All right. Here we've got 4 cubic centimeters of hydrogen peroxide, and we are putting 2 cubic centimeters of water to dilute it. What will it become? All right. Here we have 6% of hydrogen peroxide, and we are putting 0 volume of water, which means we are not putting any water, and it will remain 6%. It will remain this 6%. Okay. Calculate the percentage concentration of hydrogen peroxide solution for test tube B. Now, this is a bit of chemistry. All right. Concentration. Let's look at it this way. 6% is corresponding to 4 cubic centimeters. All right. What if we add 2 to 4? It becomes 6. Okay. This plus that becomes 6 cubic centimeters. What about if it's 6 cubic centimeters? What will it become? It will become a lesser percentage because we are diluting, so less. Therefore, percentage concentration is equal to, since it's less, the smaller number on top, the bigger number on the bottom, times percentage, 6. So this will cancel that, we'll get 4%. So here we should have 4. Calculate the percentage concentration of hydrogen peroxide for test tube B. Record your answer in table 1.1 and table 1.2. Table 1.1 is this one, we put 4 there. Where is table 1.2? Table 1.2 is this one. Okay, percentage concentration of hydrogen peroxide, we need to put a 4 also here. So, important thing, alternative to practical is about following instructions. Once you follow instructions, record your answer in table 1.1 and table 1.2. If you leave that, if you just record it here and not on the other, this homework is bound to go. All right, so carefully go through stuff. Okay. The student, step 3, cuts 3 1mm slices of potato from a potato cylinder as shown in fig 1.1. Okay, cutting. Step 4, drops 1 potato slice in each labeled test tube and starts the stopwatches. Step 5, records in table 1.2 the time taken for each potato slice to rise to the surface of the liquid. Right, so what is happening now? 3 slices are cut and they are placed into the beakers where we have different concentration. Okay, so we have our 2% here, our 4% here, and our 6%. So once we drop this, we will see this potato going to the bottom, then starting to rise to the top. Go to the bottom, then starting to rise. The time for it to rise to the top is what we are recording. All right. Let's see what next instruction records Step 5 records in table 1.2 the time taken for each potato slice to rise to the surface of the liquid. All right. The times for each potato slice are shown. Okay, test tube A as this, test tube B as this time. Now, this is a bit of physics application. Can you read time? All right, can you read time? Record in table 1.2 the results shown in figure 1.2. So we want to record these in this table. All right. Okay. Time taken for potato slices to rise to the surface, seconds. So the key is seconds. We are given our time in minutes and seconds. Minutes and seconds, 8 seconds. So no change. All right. Then we have to change to seconds. Can you convert time from minutes and seconds to only seconds? A. Let's calculate for A. 3 minutes. 54 seconds. Okay. 3 minutes in seconds, 3 times 60. Since every minute is 60 seconds. Okay. Which is 180. All right. Plus, this is 54 plus 54. 0 plus 4, 4, 8 plus 5, 18, 3 carry 1, 2. 234 seconds. So here we should write 234, converting the time to seconds. C, let's do for C. For C, it's only 45 seconds. So we put the 45 there. Okay. State a conclusion for these results. So what can we say? How are these things varying? As concentration of hydrogen peroxide is increasing, the time is reducing. Okay. So we can say the higher the concentration of hydrogen peroxide, of hydrogen peroxide is, what are the time? The time taken by the potato to rise. Okay, because we can see clearly if concentration increases, the time is becoming shorter to rise. Okay, all right. Let's go on to the next question. State and explain one. State and explain one safety precaution taken when cutting potato slices. Okay. State and explain one safety precaution. Cut away from fingers. Okay. Explain. State cut away from fingers and explain One safety precaution. To prevent adding foreign material. To the experiment. All right. Now, if you cut yourself, all right, you might add some foreign materials, say skin from your hands, or even hurt yourself, skin from your hands into the experiment. Now, the experiment might be enhanced or diminished by foreign material. So you must cut away from your fingers, okay, so that you don't add any foreign material to the experiment. Okay, identify two variables that are controlled in this investigation. All right, let's look at here. 1 millimeter slices of potato. So we are maintaining the same size, because when we talk about control, what are the things that are kept constant in A, B, and C in all the beakers? So the size of the potato. Okay, size of the potato, or we can say volume of the potato, right? What else is being kept constant? Let's look at our volumes. So we are having 2 plus 4, which will give us 6. 4 plus 2, which will give us 6. 6 plus 0, which will give us 6. So the total volume of the hydrogen peroxide solution is being kept constant. All right. So here we can say, let's just say, volume of hydrogen peroxide solution. So the concentrations are different, but the volume is the same. Okay. Suggest why the experiment is not done using 20% concentration of hydrogen peroxide solution. So you see here the time taken, look at how it is coming down. If the concentration is 2, it's 234. If it's 4, it's 150. If it's 6, it's 45. What if it's 8? It's supposed to be a very low number, like maybe 2 seconds. Now when it's 20, it will be immeasurable. All right, so the time at 20% is too short to be measurable. Time at such a high concentration may too short to be measurable. All right, we would want to measure something like 5 milliseconds. All right. Predict how the results will change if the potato slices are cut at a thickness of 2 millimeters instead of 1 millimeter. I would suggest that the time to rise to the top would be greater. The top would be greater. Why? Since mass has increased. Since mass has increased, if we are cutting 2 millimeters, we're putting double the mass. All right. We are doubling the mass. So for it to rise to the top, it will take some time because it's a larger mass. All right. The student then adds the remaining potato cylinder into some iodine solution. Okay. State the nutrient tested for with iodine solution and state the observation positive result. Iodine solution it tests for starch. All right. Observation for a positive result. Blue, black. All right. So we should see a blue-black color. All right. The students test some potato with Benedict solution by following the procedure shown. Okay. Testing reducing sugars. Step 1, put a piece of potato in a test tube. Add 2 cubic centimeters Benedict solution. Shake the test tube to mix. 4, observe the color after 3 minutes. State the instruction for step 4. With step 4, you must heat the mixture. Over a water bath. All right. Over a water bath. All right. Heat the mixture over a water bath. So when doing Benedict's test, after shaking the test tube, you heat it over a water bath, then observe the color change. State an observation for a positive result. You might get a red or orange color. Okay, those are the colors you are supposed to get. All right. And that is it for this question. The question is done. So, food tests, experimental results, control, reading time, converting time, precautions in a experiment, and concentrations. So this is what we are engaged in. Join us at one-on-one maths private tutors as we gear up for the November exams. We are revising past papers. Right now we have started on alternative to practical paper, a paper that carries a quarter of the total marks. So we have to get it. All right. Thank you very much, people.